Thank you. I'm very excited to be here to present my work and also to be a visitor in this program. I've been having wonderful time. So I'm going to talk about, I wanted in this talk to try to explain to you what my project is here at OIST during my visit, but this project is very preliminary and there aren't many results. So I'm going to spend almost the entire talk about work that we have done, which kind of gives us, puts us in a good position to study camouflage. So the first part, which will be almost the entire talk, is about predictive coding. This is, and I understand that most people here might not be neuroscientists, so I'm going to give an introduction to what that is. This is work that was led by my former PhD student, Bin Wang, in collaboration with an experimentalist at NYU, David Schneider. And then in the last few minutes, I'm going to explain how the work that we did on predictive coding makes us able to ask questions about how animals hide, how animals actively hide by generating camouflage patterns. And this is work that I'm doing here. There's a fantastic researcher here, Sam Reiter, that has done some of the experimental work that I will mention towards the end. Okay, so what is predictive coding? These giraffes here are chewing some leaves, and when the giraffes are chewing leaves, the fact that they're chewing those leaves makes a sound, and we know from decades of research in neuroscience that the brain represents external stimuli that the animals are experiencing. So predictive coding is the process of changing the representation of stimuli that animals experience in the external world when those stimuli are predictable, in this example, based on the fact that the animal knows that it's chewing. And so why is this an important thing? Because if there's a lion here, and these animals, when they chew the leaves, they want to be able to suppress the representation of the sounds that they are themselves generating, so that they are more sensitive to the sounds that this lion is making, and then they will be able to escape and save themselves. So predictive coding is changing of the representation of stimuli based on the fact that they are predictable, and this is important not only for escaping predators, it's important for learning and many other things, and there's in neuroscience a very extensive literature of this thing, which I'm going to talk about some of the important parts of that literature. Okay, so predictive coding is the phenomenon where stimulus predictability modulates the neural representations of those stimuli, and giraffe neuroscience is not a thing yet. So people study this in mice and other model systems. So in this example experiment, the animal is trained to expect a particular sound after it presses a lever. And then people record a neural activity from the auditory cortex of these animals, and they can compare the representation of the sound when that sound is predictable and when it is not predictable. And so what they find is that when the animal is not pressing the lever, this particular neuron has a strong response in terms of spiking activity when it hears that, when the animal hears this sound, and when the animal hears the same sound, but that sound is predictable based on the lever press, then that sound in the same neuron elicits a much weaker response. Okay, so this is, we can call this predictive coding for the purposes of this talk. So the next question that people are interested in asking is, what are the neural mechanisms? How does the brain make a computation? How does the brain know to expect a certain sound? and to suppress the representation of that sound when it is predictable. Before I will talk about mechanism, let me talk about this important question, whether predictive coding is stimulus specific. Sorry. So if we go back to the giraffe example, then when they chew, then that makes a particular sound. And the question about whether this predictive coding is stimulus specific is essentially, is the suppression of the sound that they make when they chew leaves, does that also suppress the sound that the lion might make? And again, this is an important question. They want to be able to suppress only the sound of chewing leaves, otherwise they wouldn't be more sensitive to the sounds of the lion. So again, this was studied in mice in the lab of our collaborator, David Schneider, and they showed that the modulation, the negative modulation of the sound that was learned to be expected is much stronger than the negative modulation of a sound that is a probe sound that was not the sound that the animal learned, follows the lever press. There still is a negative modulation, but it's much weaker than the negative modulation of the expected sound. So based on this finding, we can say that predictive coding is stimulus specific. So in other words, the brain makes a prediction for a particular sound that follows the lever press, or in more generally, the motor action. Okay, so now we get to the question about mechanism. So the question is, how does the brain compute those predictions which are necessary to suppress responses to expected stimuli? And we know based on the previous slide that this is stimulus specific. So people have been working on this for a long time. And a recent paper from Georg Keller's lab has proposed or argued that predictions originate in other cortical areas that are exchanged via long-range cortical projections. Okay, so let's understand what this sentence means. What they are saying is that there is some sensory information that enters the brain through the ear, and this sensory information arrives in the auditory cortex, the brain region that represents sounds. And in parallel, the motor cortex, which has the information about the movement that the animal makes, the motor cortex sends a long-range projection, which contains the information about the sound that is expected. And then in the auditory cortex, there is a computation which subtracts the predicted sound, the representation of the predicted sound, from the representation of the stimulus itself that the animal experienced. And by looking at this difference, you can tell whether the stimulus was expected or not expected. And they further say in this paper that prediction errors are thought to be computed in a dedicated population of neurons referred to as prediction error neurons. So in the auditory cortex, they're saying that there is a special subset of neurons that I'm showing here in with dashed lines, which represent the prediction error, the difference between the actual sound and the predicted sound. And those neurons are different from representation neurons that represent the sound regardless of whether it's predicted or not predicted. And it's important that here they say that those computations are done via long-range projections and that they are separated into these two different groups of neurons. And so let's try to think about this more carefully. So in the brain, in addition to sounds being predicted based on motor actions, your brain also makes predictions for sounds based on visual input. So presumably the same kinds of computations are done when you meet a friend, for example, and without that friend starting to talk, you already know what what you expect them to sound like, and maybe if their voice is tired, then you might be surprised by the sound of their voice. So we know that both from humans and in experiments in animals that the brain makes predictions for auditory stimuli based on visual input and vice versa. Yeah, of course. In this, yes, so far I'm presenting the kind of very common view in the field, and I'm going to talk about what we think is different in
even in the first slide in experiments, so you compare the kind of something and the movement. But the prediction itself may not require motor movement. And it's confused. Why motor cortex cognitive?
So
maybe.
I mean, people in this paper, they showed that inactivating the visual cortex leads to the inability of the auditory cortex to suppress responses to expected stimuli. So this is why they argue that this depends on these long-range cortical projections. But it's possible that some stimuli might be predictable, for example, based only on the stimulus statistics or something else, and then that might not require, the motor cortex.
Okay.
Yeah, and please, if there are other questions, I'm happy to answer during the talk. Okay, so visual information might be predictive of auditory information and vice versa. Another interesting example is visual information that is predictable based on motor information. So you experience this, for example, when you're sitting in a train and the train starts to move away from the station. Sometimes it's unclear whether your train started to move or the other train on the parallel track started to move. The fact that you're confused is because the movement was not generated by you. It was generated by something else that you might be unaware of. When you are the one generating the movement, then it's very easy to differentiate between visual flow that results from the world moving or visual flow, the same visual flow that results from your own movement. So we know, again, both from humans and from animal experiments that motor actions are used to make predictions for visual inputs. And a last example that I want to give is somatosensory touch information that is predictable based on motor input. So this is studied again in rodents that they sweep their whiskers and they learn to expect a pole that is there, which is hit by their whiskers. But in humans, a nice phenomenon that shows this is the fact that you cannot tickle yourself. So the fact that you cannot, and there's actually very beautiful papers on when you are able to tickle yourself and when you're not able to tickle yourself. So you're not able to tickle yourself because when you try, then those movements are highly predictable. So they don't surprise your touch receptors, and then you're not tickled. Okay, so I gave a few examples of predictive coding phenomena in our brain and in the brain of animals, and this picture starts to become very complicated. And so we think that this idea that predictions for every kind of sensory information are relayed via long-range projections in our brain to every other part of the brain, that seems like a too complicated way of computing these kinds of predictions. So when we started this study, we believed, based on reviewing the literature that it is unlikely that every part of the brain computes a prediction for the kind of sensory information that every other part of the brain is going to experience. And so we wanted to think about a model or a picture, a model that would be able to explain this multimodal prediction in a way that is more simple. Another problem with the current view is that so far I've talked about one motor action, which is predictive of one stimulus feature. But of course, you can imagine a scenario where different motor actions are predictive of different auditory stimuli. And if you study the mathematical models that people have constructed to explain how this subtraction of predictions, the subtraction of predictions from sensory information is done, then those models are difficult to generalize to a scenario where many motor actions are predictive of many different sounds. And so the goal of our study was to try to solve these two issues to understand how a simple network can compute multimodal predictions, and we want those predictions to be high dimensional in the sense that many different motor actions will be predictive of many different stimulus features. Okay, so here's a schematic of a recurrent neural network that we built that can do this. So this network receives a high dimensional array of auditory inputs that are that we call x1, x2, all the way up to xp, and there are corresponding motor commands. Y1 is predictive of x1, y2 is predictive of x2, and so on. And so there is some connectivity here, which I will talk about briefly in a moment. But what we want this network to do, we want this network to do some kind of processing such that from the activity of the neurons in this network, we are able to read out the stimulus identity, meaning was the animal presented with x1 or x2. We want to be able to read out a prediction, meaning that if y1 is presented, we want to be able to read out a prediction for x1. And we want to be able to read out a prediction error such that if y1 was presented and x1 was not presented, we want to be able to say that based on the neural activity in the network. So it's very common in computational neuroscience these days when you want to solve a problem like that to ask a machine learning algorithm to solve that for you. And that kind of approach has some advantages, but it also has some disadvantages. So we used a different approach. And so we used a prescription for the connectivity. We wrote down an equation, a formula for the connectivity in this network such that those readouts are available. And then we, because this is a prescription, then we can investigate under what circumstances those readouts are available or not available. And this is similar to this prescription that we used is similar to what was previously proposed by other authors, but from a theoretical point of view, the novelty of our study is that we can analyze this network in the high dimensional scenario. And here I really want to acknowledge the work of my student, Bin, that really did beautiful mathematical analysis of the system. The network units are nonlinear. Like we know that neurons in our brain have a nonlinear input-output transformation. So we want to be able to analyze the activity of this network when the neurons are nonlinear. and our results show that the fact that the neurons in our network are nonlinear is very important for what the computations that this model does look like. And then the final thing that I'll touch upon at the end, if I have time, is that the network units can be separated into separate populations that are excitatory and inhibitory. So we know that in our brain, we have different groups of neurons that some of them are excitatory and others are inhibitory. And so we started with a network that violates this, and then we're able to derive a network that respects this separation. Okay, so a cartoon of what we want the model to do if we have a stimulus space with inputs X and Y. In the mismatch condition, X is large and Y is smaller, vice versa. And in the match condition, when the stimulus is predictable, then X and Y are either small together or large together. Then the stimulus is somehow transformed by the neural activity in the network, and we want this transformation to be such that we can draw a hyperplane which separates all the stimulus conditions that are mismatched from those that are matched. And if we project on a line perpendicular to this hyperplane, we will have strong firing rate responses in the mismatch condition. Those are prediction error, like we saw in the experiment. And in the match condition, the responses will be small when the stimulus is highly predicted. Okay, so I'm not going to go into the details, the mathematical details of this model, but what I do want to explain is what is the mechanism that allows this network to compute predictions. And that mechanism is a balance between the input that every neuron receives from the outside world. So every neuron receives information from the outside world. This information can be both motor and auditory in our example, and it receives input from other neurons in the local circuit. So we assume in our model that every neuron tries to balance these inputs. Every neuron tries to have a balance level B, which is high, such that the feed-forward input is large relative to the difference between the feed-forward and the recurrent input. And so when the model learns to generate those predictions, in the match condition, the balance level increases because the network learns to generate recurrent input, which more closely tracks the feed-forward input. And in the mismatch condition, the balance goes down, which generates those large surprise responses. And late in learning, the distribution of those balance levels for every neuron in the mismatch and match condition are highly separable, so we can put that hyperplane that I had in my previous slide. Okay, so what we found by mathematically analyzing this network is that there's an optimal balance level, B star, which gives us strong mismatch responses, which we want because animals want to be able to have large sensitivity to surprising stimuli, and at the same time have suppressed but non-zero match responses. And this is a cartoon of how we do this optimization. We want suppressed but non-zero match responses because we know that even when the stimulus is totally predictable in this experiment, the response of the neuron to this totally predictable stimulus is not zero. There is still some weaker but non-zero response to the stimulus, which is predictable. Okay, so this is all this picture is for a model that is trained to make one prediction, a prediction for one stimulus based on one motor action. the next thing we did is to ask what happens when we now increase this parameter P, which is the number of stimuli, and the correct scaling is P relative to N, which is the number of neurons in our network. So when P is proportional to N with proportionality alpha, then the optimal balance level, this was a surprising result to us, the optimal balance level, how closely every neuron is trying to match the feed-forward and the recurrent input, goes down. So you're trying to learn more and more of these associations between motor actions and predicted stimuli, but to us counterintuitively, at every neuron, the match between feed-forward input and recurrent input actually goes down. And so the intuition, after thinking about it for a while, the intuition why this still works is that when the balance between feed-forward input and recurrent input is tight, meaning that they track each other very closely, then predictions for different stimuli interfere with each other more. And so this creates a lot of noise in those predictions. And so when the balance level is lower, that noise from interference between stimuli is weaker, and the predictions are more accurate, actually, when the balance is lower. And a prediction that comes out of this result is that when animals are trained in enriched environments where they play around and different motor actions lead to different sensory consequences, we would expect the balance to be more loose in the brain. And the balance has been measured in the brains of animals since the 90s. There's a rich history of trying to understand the effect of balance between excitation and inhibition in the animal brain. So if you interpret the balance between feed-forward input and recurrent input in our model as not balance between feed-forward and recurrent input, but balance between excitation and inhibition, then our model can explain why different people report widely different balance levels between excitation and inhibition in the brains of animals. The explanation would be that they might be raised in more rich environments where they are, their brains are capable of making predictions for a higher dimensional stimulus set. And again, I'm going to make this connection between balance between feed-forward and recurrent input to balance between excitation and inhibition towards the end. Okay. So moving on, what do the model's predictive representations look like? Like we think we have a model that explains how the brain makes predictions for many different stimulus features. If we now stick an electrode in an animal's brain, what do we expect? And that animal is trained to make predictions for many different stimuli based on many different motor actions. What should we expect to see? And is it true, like was proposed before, that there's a separate population of neurons that represent the stimulus, regardless of whether it's predicted, and other neurons that represent the stimulus, that represent the prediction error? So to answer this question, we need to be able to classify the neurons in our model. So we use classification that is very common in the literature. When the neuron firing rate increases as a function of X independent of Y, we call this a representation neuron for X. And similarly, a representation neuron for Y doesn't care about X. And a prediction error neuron is proportional to some nonlinear function of the difference between Y and X. So the first thing we can show, not surprisingly, as the model learns to make predictions, then the fraction of neurons that are prediction error neurons increases. Again, this is not surprising because the network learns to make predictions. The interesting thing is what happens at high dimensions. So what we do here is we compute this quantity delta. For every neuron, we compute this quantity delta, which is the difference between the mismatch and match response to a particular stimulus. And so we do this for an association between one motor action and a particular stimulus, and then another motor action and a different stimulus. And so if neurons are in these light blue rectangles, this means that they are representation neurons for one association and prediction error neurons for another stimulus association. And based on the previous proposals, these neurons should not exist. And we see that when the network learns only two of these associations, then there aren't many. There are some, but there aren't many of these neurons that have a mixed representation of faithful representation of the stimulus and a representation of a prediction error. But when P is proportional to the number of neurons in the high dimensional limit, which is what we expect to be the case in natural conditions, then we see many more of these mixed representation neurons that are in these blue rectangles. So we can plot the fraction of neurons that are in these blue rectangles, and it increases about twofold when the stimulus dimension increases. So this should not be the case if you go by the previous theories for how networks compute prediction errors. Okay, so this is quite different from what was proposed previously. So we want to test this idea that predictive representations are functionally desegregated, that there's a mixing between representation of the stimuli and the representation of the prediction errors. So thankfully, our colleagues did this nice experiment where they trained the animal to press a lever and expect a particular sound, but then they violated the animal's expectation of this particular sound in 4 different ways. They played a different sound. They played the same sound but louder, the same sound but more quiet, or the same sound but from a different spatial location. And we can compute this quantity delta for the association between the motor action and either the probe sound or the expected sound. And there's a subtlety here that I don't want to get into, that the motor action is actually the same motor action. So this is not the perfect experiment to test our prediction, but this is the experiment that our collaborators did. So if the representation of the stimulus and the prediction error is segregated, like was proposed in previous works, then we should see a strong correlation between those two quantities delta. And if they are desegregated, then we should expect a small correlation. So we called the correlation between these two quantities delta, we call this a segregation index, which is equal to 1 when those representations are segregated. And we plotted this segregation index as a function of the similarity between the two stimuli in terms of their representation in the animal's brain. Importantly, this was computed when the animal heard those different sounds without pressing the lever. So this is in the passive condition. And we see that when the sounds are different enough, particularly when the animal was probed with a surprising stimulus with a different frequency, then the segregation index drops quite significantly. And this is very different from what was expected, but based on previous literature, that the segregation index will be always high and independent of how similar the stimuli will be. So we think of this as some evidence that when you go to high dimensions, the representation of stimuli and prediction errors in the brain are mixed in the brain of animals. Okay, now a very important question that we wanted to address is how realistic is the model we have so far? So we have a model, we can do a mathematical analysis of the different properties of this model, but it is a model, and we don't know if the model is able to reproduce the properties of prediction error neurons in the brain just by chance, or because this is really how networks in the brain compute predictions.
Sorry.
Yeah.
I missed it. What is smallest in the PDSP?
Oh, sorry. There is one parameter. There's one parameter in the model that I didn't tell you about, which is how sparse the neurons are, how likely a neuron is to respond to a stimulus by chance. So this is like a sparsity parameter, and we saw that the same model for different levels of sparsity. This is a parameter that we can't really fit based on data. So we show that for reasonable values of this parameter f, we get a reasonable match to the data.
This means if the representation is sparser, segregation is lower. The segregation, I guess, is between sensory neuron and
yeah.
So you have a smaller number of prediction error neurons. You know?
When the representation is more sparse, then I'm not sure I want to make a big deal out of it, because when the representation is sparse, then in general neurons are less sensitive to anything that's going on. So I'm not sure.
Okay, so rather opposite. So two sensory and
they might
neurons are more mixed.
Yes. Yeah. Okay. Okay. So recent work in this field of predictive coding suggests that the brain is not equal to some homogeneous soup. What do I mean by that? I know that this is a very controversial statement, but so in this paper by Gerard Keller, they show that neurons with different molecular identities have different functional properties in terms of how they represent stimuli, whether they are more likely to represent the stimulus or the prediction error. And in this paper from the lab of Tom Mercier-Flogel and Sonia Hofer in London, they show that not only within a specific region that was studied here, but if you look at across the thalamus and the cortex, then different regions of the animal brain have different roles in computing these sensory prediction errors. So the problem that we wanted to address is the following. So we have this model, and I told you that this model can reproduce some of the properties that we see in real neurons that are recorded in prediction error experiments. Now, if you look at the synaptic connectivity matrix of this model, it might look something like this. And if you look at a single column of this matrix, meaning the connections from a single presynaptic neuron, you see that they can be both red and blue, meaning that this neuron in our model is both excitatory and inhibitory, which is not true in real neural network. So it is similar to saying that we are assuming in the model that I told you about so far, that the brain is like a soup, homogeneous soup. It's a little spicy because our model can do interesting things, but still it's a soup, which is not good. So we wanted to fix this problem. The way we fixed this problem, so we wanted to unsoup the network. And we did that actually, the inspiration for this came from a paper by Tomoki. We looked for a way to separate this connectivity matrix into 4 different modules. And we did that using non-negative matrix factorization. We were able to do this analytically, again, because we have a specific prescription for the connectivity. We don't use some result of machine learning optimization. And so you see that after we do this non-negative matrix factorization, every column of this matrix is either blue or red. So now our network respects the fact that every neuron in the brain is either excitatory or inhibitory. And we get 4 different components for the connectivity. We get connections from excitatory to excitatory neurons, from excitatory to inhibitory, from I to I, and from I to E. And when we did this non-negative matrix factorization to get those 4 components, what we found is that this decomposition into 4 components is not unique. There is a 1-parameter, we were able to find a 1-parameter family of networks that all reproduce the nice properties of this network in terms of prediction error responses. But the structure of inhibition in this network is still kind of a free parameter. So on one end of this line, we have inhibitory neurons that are private to every excitatory neuron. So every excitatory neuron has its own private inhibitory neuron, which flips the activity. This is a trick that many theoretical studies use. And in the other end of this, line, we have inhibitory neurons that are not private to a particular excitatory. We have inhibitory neurons that are not private to a particular excitatory neuron. We have inhibitory neurons that are sort of private to a specific stimulus. And we have everything in between. Everything in between will give us this nice network that we started with. So what we did is to infer this parameter lambda from data where our colleagues recorded in those prediction error experiments, they recorded separately inhibitory neurons and excitatory neurons. And based on this analysis, we were able to show or to estimate that lambda is about 0.6. So there is some mixture of connectivity structure that is more private and a mixture of connectivity structure that is more an internal prediction where every inhibitory neuron is specific to a stimulus. So we made, and we think, an important step towards getting a realistic model that has those interesting capabilities of computing sensory prediction errors. And an important conclusion that came out of this is that counter to what was previously proposed, learning learning to make predictions does not simply potentiate or increase the strength of inhibitory internal model. It's not that when I showed this cartoon where the prediction is subtracted from the external stimulus, this subtraction is not simply, let's increase all the inhibitory connections and they will represent the prediction. We see that by looking at the distribution of connections from inhibitory to excitatory neurons before and after learning, and we see that many of these connections actually become weaker. So inhibitory and excitatory neurons in our model, they somehow work together in a complicated way to compute those predictions. And based on our model, we think that we can now, if people record those experiments, those neurons in additional experiments, we can test the predictions of our model. Okay, so let me summarize the first part of the talk. We propose a network model that has desegregated error and stimulus representations. In our model, predictions are computed internally in the local circuit. They don't require explicit learning of some internal model in a faraway region of the brain or a specific network architecture. So what we're proposing is that the motor cortex can broadcast information about motor activity, which will arrive in the auditory cortex. And the auditory cortex, through the kind of network that we are proposing, will be able to make the necessary predictions of what sounds are expected. So this is a much more simple picture for how the brain can compute multimodal predictions compared to what was proposed in the previous literature. When the stimulus dimension becomes large, our model predicts that the balance between excitation and inhibition will be loose, and we will have more desegregated neural responses, a larger mixing between representation of the stimuli, which is independent of motor action, and representation of prediction errors. And we mapped our network model to a family of networks with separate excitation and inhibition, and by fitting this parameter or estimating this parameter based on data, we can argue that predictions are not simply potentiated inhibition. Okay, so in the last 3 minutes,
so you're proposing that for every sensory system, you would have a local model for the prediction of.
Exactly, and our model, after we had those results, we read back some of the literature, and the postdoc work of our collaborator, David Schneider, they had a quote from in a paper from 7 or 8 years ago, where they said that it's interesting to understand how promiscuous connectivity from the motor cortex to the auditory cortex could be used to generate those predictions. And we think that exactly this promiscuous connectivity, our model explains exactly how such promiscuous connectivity could be used to do that. Yeah.
I have also the other question. I'm a bit confused that this is for the learning-related behavior, or you're also thinking about the inference copy ideas?
So it's a good question. So the model that we studied so far, there is a parameter which describes learning. The network connectivity changes as the animal learns to make those predictions. There are, and the experiments that we're modeling, surprisingly to me, it takes an animal about 3 to 4 days to have 3 to 4 days of training to have reliably suppressed responses to predictable stimuli. There are other experiments where representation of sensory information is suppressed based on its predictability in a way that and those predictive representations emerge over a minute, not 4 days. So a very classical experiment is called oddball, where you present it to an animal a series of beeps at 1 hertz, let's say. And so the animal hears beep, 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 beep. And then after the 3rd or 4th beep, the representation of that sound goes down. And when that beep is omitted, there's a prediction error. So that can be learned within a minute. And And we think that our model might be a beginning of an answer to how similar prediction error responses could be learned on a time scale of 1 minute or on a time scale of a few days. But you had a follow-up question?
Yeah, but for here, like, what do you say is the motor cortex to the auditory
Yeah.
cortex connections, right? So to that one, if we were training from the river press to the other specific sound unit associations, but the previous experiment which David Schneider did with the Rich Moody was that the footsteps, right? They have to hear the footsteps more
Yeah.
like a chewing of the leaf you are explaining
Yeah.
that one. That is more like their own movement, right? They don't need to learn the association, rather it's they are hearing their own movement sound. That case they are using.
Why are you saying that this is not learned? I mean, maybe
Yeah,
it's learned just very early on in development.
Yeah, both of the case is that the predictions, but a prediction probably slightly different based on the behavior. That's the thing. I'm kind of asking that whether which both of the case there you think it's using or they're.
I think I'm not sure I see the distinction, but maybe this is something we can talk about later. I wanna talk for 3 minutes about what I'm doing here, and then I'm happy to take more questions. So camouflage. In camouflage, animal actively generate a motor command to hide themselves from predators and prey. So this I took these 2 snapshots from a YouTube video, but you're fortunate enough to be able to see these things in your backyard. So this is an octopus, and here there's also an octopus, and these 2 snapshots were taken 2 seconds apart in this YouTube video. And the question is, how does the brain of this animal compute a motor action, which is high dimensional, which is enabling it to hide itself completely from an external observer. And so Sam Reiter, when he worked in Gilles Laurent's lab in Frankfurt, they published this paper in 2023. So the skin pattern that this animal generates using chromatophores is high dimensional. Here they project it on 2 dimensions, and they in 2 dimensions they have this undulating path from the start point when the animal is not well camouflaged to the end point where the animal is well camouflaged in its environment. And what I think is the most interesting result in this paper is that if you compute the angle between, so in these dark regions, the speed of changing the skin pattern, this is not the speed of the animal moving, this is the speed of the animal changing its skin pattern in high dimensions. That speed, when that speed is low, you can compute an angle alpha between the direction from that slow region to the endpoint and from that slow region to the next slow region. And what they show is that that angle alpha is much closer to 0 than you would expect by chance. Meaning that a finding is that after or during those slowdowns, the skin pattern accelerates in the direction of the endpoint. So now we want to use our model to interpret this finding and try to understand how the brain generates this behavior. So I replaced now the visual input, we believe, are predictive of the animal's motor commands. And again, this is in high dimensions. So our interpretation is that at the slow points, the animal predicts the appropriate motor command based on the visual input. And this is why the angle alpha is close to 0. A question is, how do these animals know how to stop? What feedback mechanism do they use to stop. We know in which direction they exit the slow points, but we don't know what makes them stop at these slow points. So using the same cartoon or drawing a new cartoon on this data, if we assume that this angle alpha is minimized based on a prediction that is computed by something like our model, our idea is that we can use the fact that this model computes prediction errors to know that this angle is in red between the direction that it's going and the direction of the endpoint. That angle is a prediction error. And when that angle starts to become large, then the animal should know to stop and recompute the prediction. So at the discussion of this paper, they proposed 3 different mechanisms that could implement this feedback computation, proprioception, vision, or inference copy. And so our guess is that the animals use visual information to read out the prediction errors from specific linear combinations of the chromatophores that are accessible to them through the visual system. And with our model, we have the kind of tools to test this hypothesis. So I just want to thank primarily Ben, who helped or led this project, and this would not have happened without his very hard work, and David Schneider, who gave us the data and was a wonderful collaborator. I want to advertise that there are wonderful people here. If you're interested in coming to another place on the other side of the Pacific where people do interesting work, then my lab is looking for postdocs. And I want to again thank OIST for hosting me during this visit. Thank you very much.
Have several questions. With mine?
Yeah, of course.
Yeah. So, yeah, remember your model is a concept of adaptable to do with.
So our model is not exactly an attractor network. All the mathematical analysis that I didn't show is done at steady state, in the sense that the 2 stimuli. So we are cheating a little bit, and we are assuming that the 2 stimuli are presented for a long time, such that the neural activity reaches steady state. Now, a reviewer of our paper asked exactly this: what happens when you present the stimuli transiently? Because we know that when the stimulus is predictable, let's say 1 second after the lever press, then that's transient activity, that's not steady state. So we are now investigating how the predictive representation of our model looks during transit.
Actually, my question is slightly different. Even if you use, so I wonder if you increase the number of patterns or stimuli, and there will be some critical point where, for alpha, where beyond that prediction is not sufficient.
Yeah, so this is something, it's not, so this model doesn't have the same notion of capacity as a Hopfield network. It will stop working actually for reasons that are not all the way clear to me. For alpha greater than 0.5, this picture breaks down, but this is not the same catastrophic forgetting mechanism as you have in a Hopfield network.
Just second order transition. Okay, sorry. Okay.
I mean, I don't know, yeah.
Is there any other question?
What's the difference between, like, how do you tell if the real system is doing what the experience doing by like memory, like this is pricing based on memory versus I'm predicting something and that mismatch with like prediction. Like, is there a way to tell the difference between the mechanisms?
So usually if it's memory-based, people would expect this to be something that is emerging on longer timescales than if it takes, let's say, 30 minutes for synapses to change, and the basis of the memory is synaptic plasticity, then maybe we can call something memory if it takes more than half an hour or an hour to emerge. Now, this is a very problematic statement because there is also short-term synaptic plasticity. So I think that that it's very difficult to differentiate between these two possibilities.